So I'm delighted that we can present this revised Annex 1 information session. My name is Gordon Farquharson, and I'm one of the initial group who started up the Contamination Control Network. We decided that this was an important subject following the publication initially by the European Commission in August this year, 22nd of August, and subsequently the Pharmaceutical Inspection Cooperation Scheme published their version. And the two are identical texts. I want to make a comment, the green note at the bottom, that I'm providing some factual information about the content of this revision, and then I'm recording and reporting some industry-related feedback and concerns, and also my own personal professional opinions on some of the issues, and also on some clarification that I am providing under the banner of the Contamination Control Network. It is early days, and the industry is working hard to get its arms around a substantial revision of this regulatory guidance. It has moved from 15 odd pages to more than 50 pages, and clearly that doesn't happen without a significant change and increase in the guidance included. This presentation is going to focus on the clean room and clean zone related aspects. This regulatory guidance includes a great deal in addition to this related to various aspects of sterile products manufacture, including specific guidance related to certain aspects of the manufacturing process. Now, we're not going to have time and no intention within this session to get into every particular, so we decided to focus on the clean room contamination control related aspects. And I'm delighted to welcome you all to this. We've had an amazing response, and I hope that this presentation gives you at very least a heads up on major changes and some important aspects that we need to clarify and develop best practice related to. So a deadline. There are two deadlines published on the front page of the European version. So 1 year, so 25th of August 23, for a majority of the document to be enforced, and 2 years related to some specific aspects of lyophilizer, sterilization and utilization. Within this presentation, I have tried to differentiate specific text from the document, which is in blue italic script, and then most of the comments and observations are in dark green regular script. So why was the document updated? Well, that is actually published very clearly in the foreword, so it is worth just reflecting on the rationale and justification. So there was a joint inspectors working group from the European Medicines Agency members and the Pharmaceutical Inspection Cooperation Scheme, and they worked together on this revision. Now, this is the first time that there has been a significant regulatory document of this sort that has actually been written in a cooperative way. Historically, PICS, Pharmaceutical Inspection Cooperation Scheme, has taken on board and endorsed or engrossed the European Union GMP as the PIC's GMP with some small modifications. It's very, very important to recognise and understand that in PIC's member nations, the GMP from PICS is implemented at different speeds. Now, this generally is because in many nations, the pharmaceutical good manufacturing practice guidance is embodied in some legal vehicle, and this often takes additional time. And it's important to remember that in the European Union and those countries that are associated with the European Medicines Agency, that very often and usually the GMP guidance sits under the law, so the implementation does not require laws in Parliament to be changed, for example. So it was a joint venture, and it says, and I've highlighted some key words, the new guidelines should clarify how manufacturers can take advantage from application of advanced process understanding, enhanced process understanding, and by using innovative tools described in ICH Q9 and Q10. And it's particularly important to recognise that ICH Q9, quality management, was itself significantly revised during the time that the Annex 1 was revised. So the revision should also take into account related changes in other GMP chapters and annexes. So, for example, Annex 1 does not address anything associated with cross-contamination control. These aspects are still addressed in sections 4 and 5 of the general provisions of the GMP document. Then the final clause is that the revised guideline will seek to remove ambiguity and inconsistencies and will take account of advances in technologies. Now, as we look through the document, I think it's been somewhat but partially successful in that respect. And I personally have a number of concerns about inconsistencies and ambiguities. And indeed, those who follow the subject will know that in organisations like PDA, ISPE and PHSS in the UK, there have been significant meetings in the very recent past debating some of the issues and problems, and I'm sure that industry associations will be constructively positive in trying to help address some of these problems. So first of all, something about the language. Shall, must versus should and may. Do we understand how the language is used in the document? So there is much confusion about the intent of the word should in not only Annex 1, but generally in regulatory documentation. Traditionally, I think there was clarity. The word shall, historically, would have been used for objectives, actions, responsibilities, duties that are deemed to be unambiguous requirements for the subject concerned. Things that are compulsory, necessary. In our case, regulatory requirements. So sterile products shall be free from microbiological contamination, and this can be achieved by both aseptic processing and terminal sterilization. So that is an unambiguous requirement. How you get there, the should, was traditionally used for solutions, actions, responsibility, duties that are suggested or advised as being appropriate ways of achieving or meeting the objectives. Alternative approaches with equivalent outcomes are acceptable, but need to be justified. In Annex 1, there is no such differentiation between shall and should, so you'll see the document is all dominated by the word should. And therefore, we have a responsibility to decide if any of those shoulds are really shalls and to justify our conclusions and any alternative approaches that we care to adopt. So that is a very important principle. The document uses the word should, and within that, some of the guidance information is used to be an illustration of current best practice, but alternative approaches may be appropriate. We must always remember that our GMPs and our GMP guidance lacks best practice and technology, probably by 5 years and in some cases even longer. I'm not going to go through this in any detail, but there is a content called a document map, and that shows very clearly that the revised document contains basically the same scope as the 2008 version, which is the one that most people will be working to, but the volume has expanded and there is a great deal more depth and detail. And clearly that depth and detail has come from parties in the working group that developed this, who believe that much more explicit detail is required. So let's begin with the meat. So the first thing is the concept of a contamination control strategy is introduced. Now, I don't believe that this changes basic requirements, but effectively it is recommending a more comprehensive, coordinated and documented compilation of measures taken to control contamination related to sterility assurance. So, for example, there's nothing expressed in there related to cross-contamination control, which would be another important aspect of our GMP practice. The guidance in Annex 1 lists very clearly that the contamination control strategy will consider physical measures to do with equipment and manufacturing facilities, operational measures related to standard operating procedures, selection and training of personnel, the whole gamut of qualification and process validation as set out in Annex 15, and monitoring both of process, but in terms of contamination control, very specifically monitoring of the manufacturing environment. So it starts off with that high-level picture. The Annex 1 guidance then goes on to suggest broader scope of aspects that should be considered in the contamination control strategy. So firms will need to assess how they're going to achieve the development of their own contamination control strategy. The document does not describe the form that this should take, and I'm sure that this will be something that industry associations will provide their members with ideas and guidance on pretty quickly. But if we drill down into the text, we will see that CCS covers process plant and equipment, personnel, premises and facilities, utilities, raw materials, containers and closures, supply chain management, risk management process, outsourcing, validation and qualification, preventative maintenance, facility monitoring, and the quality management system. So all those appear in a list of the contamination control strategy should encompass the following, and then it gives a list of those. The thoughts from industry so far are that a firm is not going to be required to rewrite all the elements here, but are more likely to have some guidance document, some higher level document, which refers to largely pre-existing documents covering each of those subject areas. The challenging bit will be firms will need to decide whether they have a contamination control strategy that is departmental related, product related, or process group related. I think this will be very much dependent on the type and nature of the manufacturer's business and the range of products and processes that they have within the factory operations. Annex 1 will not be prescriptive about this, but paints a picture at quite a high level, requiring some thought. Now, in today's discussions, we will look at the things that really just relate to process plant and equipment, personnel, premises and facilities, particularly clean room and clean air devices, utilities that might impact those, and particularly clean room related monitoring. And I guess every time we talk about our current good manufacturing practices, we talk about hazards and assessment of the risk of those having an adverse effect on our products or processes. So that's always an important overlaying consideration. So drilling down now into more detail. So let's start off with the cleanliness grades. So the grades A to D, the good news is there are no changes to these references. They still apply in the form we're comfortable with from the 2008 version. But as you'll see a bit later on, there are some subtle changes to the cleanliness attributes of particularly grade A and grade B. The grades of cleanliness are applied to sterile product manufacture by both aseptic processing and terminal sterilization. And this is where the guidance is quite different from US FDA's guidance, which only exists related to aseptic processing. So the grades A, B, C and D still apply. And within the document, absolutely clearly, the main focus is on the highest risk processes, which remain as aseptic processing. So terminal sterilization is also considered, but aseptic processing dominates the guidance because the hazards to process integrity are the greatest. So I begin by thinking about what Annex 1 says about contamination control technology to use. So which cleanroom contamination control technology to use? Traditional cleanroom and clean air devices, or barrier technology which in the document very clearly include restricted access barrier systems and isolators. So these different contamination control technologies are clearly expressed in the guidance. So does Annex 1 imply, require, demand a particular technical solution? So if we read clause 4.3, we see that there is a very clear expectation. Restricted access barrier systems or isolators are beneficial in assuring required conditions and minimizing microbial contamination associated with direct human interventions into the critical zone. So in this clause we get a very, very clear pointer that barrier technologies, their principal advantage is separating or improving the separation of people, a major source of contamination, from the critical zone. So that could be the aseptic filling zone or a point where a product or process is exposed to the environment. Their use should be considered in the contamination control strategy. Well, I don't have a problem. I would say if you have a contamination control strategy, their use shall be considered. Any alternative approaches to the use of RABs or isolators should be justified. Okay, so I read that as being, well, this is now the norm, and if I wish to do something else, I'm going to have a very clear justification. Maybe it's expensive would not be an adequate justification, but maybe medicine shortage would be. I just don't know. So what are the alternatives? Are there alternatives out there? What would be a justification for not considering barrier technology or deciding that these technologies were inappropriate? I don't have all the answers, and I think this will probably be something that will be a source of further debate. Interestingly, there's lots of new guidance on barrier technologies in clauses 418 to 422. So I just had a little think and thought about what are the alternatives. So the primary objective of barrier technology, if you pick through Annex 1, are to separate people, operatives, from the process, but also clearly to provide a closed environment that can be effectively biodecontaminated by automated means. It seems these two aspects pop up multiple places in the guidance. There are some challenges with today's barrier technology solutions. Complex, or some would argue perhaps not if we have established solutions. Expensive and maybe inflexible. And flexibility may not be a driver in your particular application. And of course we also have from UK MHRA still hanging in the background about the fragility of processes such as hydrogen peroxide biodecontamination that have clearly informed, in my view, some of the hardening expectations on microbiological decontamination of isolators and restricted access barrier systems. So, what alternatives might be acceptable? So at the moment I would see two, maybe adaptable robot manipulations in very simple clean spaces with no personnel needing to be present. In fact, we might see something move a little closer to the semiconductor industry and the way that they would operate in some of the process automation cells. The potential of this would be to allow much simpler and more flexible space. then the other possibilities are probably legacy systems updated as practical, which already have proven robust performance, but perhaps with a plan as to their life cycle and some plan for retirement of these assets. As we know, assets don't last forever. Okay, so moving on, barrier technology, lots of new details in the guidance. So first, we have a term that was very, very common in my experience in particularly in Germany, in the description of clean rooms and clean air devices in VDI 2083 guidance, for example, the concept of first air. And this is now described in the little dictionary of terms, and it is clear it refers to filtered air, coming from a HEPA filter in our cases, that has not been interrupted prior to contacting exposed product and product contact surfaces, with the potential to add contamination to the air prior to reaching the critical zone. A term that is coined within the document, and it's referred to in the text in terms of the application. And there are a lot of important impacts of this related to how clean air is configured and how the process equipment particularly is configured within that clean zone. So drilling into the detail, we now see isolators divided into two clear types: an open isolator, and an open isolator is one where there is some continuous process transaction through the isolator, so feeding in of components and feeding out of closed containers, for example. In the document, this requires grade A in the critical core, unidirectional airflow, and first air is described. So the air coming from the filter source needs to touch the critical product or process prior to being contaminated from any other secondary surfaces. Now we have a change. The background minimum cleanliness is GMP grade C. 2008 it was grade D. So why has that change come about? I suspect it is part of the alignment with US FDA practice, and practice that is fairly common but not universal in current applications. So what about legacy isolator systems, that could be open or closed isolators, that are operated in grade D today? Then for open isolators, there is additional requirement for testing interfaces, particularly mouse holes for product discharge and waste streams. We then move on to isolators closed. So there's clear differentiation between closed and open isolators, and these are isolators in which the materials for the task generally are loaded into the isolator, and then there is some microbiological decontamination process. The work is carried out, and then the materials, once secure, are removed from the enclosure. Requires grade A plus unidirectional airflow and first air for processing lines, it says. So presumably for other things in such isolators that are not processing lines, first air is not so important. I don't quite understand that. Then there's a rephrasing of very unclear language that existed in 2008 version, and it says you can have grade A with non-unidirectional airflow for simpler operations. Now I really don't understand what simple operations are. I would have thought it was more related to operations that are not generators of airborne contamination, that would need to be swept away by unidirectional airflow. That's really, for me, the only justification for a non-unidirectional airflow grade A zone. Now the background environment minimum moves to grade D, and I guess you could apply that, and that would be logical related to, say, sterility testing in an isolator. Then there is a note that negative pressure isolators, only if they are essential for a hazard, and they must also protect the product. So the assumption here is that negative pressure isolators can only be closed isolators. And I'm not sure from an engineering point of view whether that is a necessity. And then restricted access barriers, the requirements are less complex, and we require grade A plus unidirectional airflow and first air protection, and the surrounding environment must be grade B. So quite a lot of new detail to get our minds around. A few other points, and again I'm not going through this exhaustively, but key points. There's much more guidance on gloves and biodecontamination. So for gloves, for example, there's more information and guidance on testing frequency, and it describes both sterilisation of gloves through a pharmacopoeia process in an autoclave and disinfection or biodecontamination in situ using some gaseous decontamination system. Biodecontamination, so it describes this as a clear objective to ensure surfaces are free from viable organisms. An automated process is advised, and then suddenly there's a particular detail about gloves should be extended. It highlights just that aspect. I think there are some other aspects related to gloves that are equally important. And then it seems to focus on hydrogen peroxide processes, and there's no mention of alternatives such as chlorine dioxide or nitrogen dioxide. So are these agents excluded? I would assume they could be included under the flexible interpretation of should, provided you have process understanding and a rationale and justification documented in the contamination control strategy. Other cleanroom features are now subject to more detailing, and I'll just quickly look at airlocks, airflow visualization, pressurization, cleanroom qualification and clean air device qualification, and ongoing monitoring. Airlocks. So there are lots more words on airlocks, and the language is intended to apply to transfer hatches and gowning rooms. It talks about, in a philosophical sense, about temporal or spatial separation of entry and exit, depending on the amount of traffic, and particularly in the context of gowning rooms, you clearly need to avoid those entering a controlled environment from rubbing shoulders with those leaving it. But it's your decision as to whether you can do that on a time management basis or you need separate in and out. It still has the terminology and the expression that gowning rooms are special kinds of airlock, but still has this strange text from the 2008 version of Annex 1, which I still struggle to understand, where it says the final stage of the airlock should in the at-rest state be of the same cleanliness grade, viable and total particle as the clean room into which it leads. Now generally, if we assume this was an aseptic clean room, a grade B clean room at rest should be largely free from viable particles. So why does it just say the at-rest state? So what does it really mean? What about the in-operation state? Gowning rooms are clearly very demanding for grade B, and this would be one of the very positive arguments for moving to barrier technology, isolator barrier technology, where we can relax the surrounding environment and release ourselves from some of these very demanding gowning requirements. Gowning guidance, so there's lots more detail in the personnel section 7.3 to 7.17, including some guidance on scope of garments donned for different grades, very similar to before. Some more stringent requirements for garment vendor, garment testing qualification, which seems to me to be a new explicit requirement. And there's some guidance on space in gowning rooms. Now, I would just add my personal reflection that gowning rooms are often poorly designed because the gowning process is indeed a process, and a complex process that enables people to move from a less clean state to a more clean state and back again. Airflow visualization. So this is in the document. It relates to both non-unidirectional and unidirectional airflow rooms. So there's more detail, but it's a very general expectation, and I believe we need to be very, very careful to review and clearly decide what we're going to evaluate. Airflow visualization is not a quick, simple, casual task. It's invasive, it's time and cost consuming, so we need to do it where it's truly necessary. The revised Annex 1 suggests that airflow should be, that's airflow visualization, air patterns should be visualized in non-unidirectional rooms. Now, this is truly difficult to do. My experience is you get very, very limited information, and you're more likely just to get a fog of smoke in a room very, very quickly. So it's difficult to do and likely, in my opinion, to provide limited information. Ultimately, the test of the pudding is that the room can maintain and achieve at rest and in operation cleanliness levels for particles and microbial contamination. That ultimately is the acid test. Airflow visualization is recommended clearly for conveyors, mouse holes and the similar, going from cleaner to less clean rooms or zones. This seems a very sensible addition to me and could be applied to clean rooms and clean air devices such as isolators and RABs. And then there is a bit more detail about visualization in unidirectional airflow zones, grade A zones, and that's clearly reflecting current established practice. Room pressure, indicators, alerts and alarms. Lots more about this than in the 2008 version, so more detail. 416, indicators of air pressure difference should be fitted between clean rooms and or between isolators and their background. Okay, it doesn't say what kind of device this should be, but we have freedom to select an appropriate device. Air pressure differences identified as critical should be continuously monitored and recorded. Well, I think it's more explicit in this version, but I would say that's largely current practice in some shape or form. A warning system should be in place to instantly indicate and warn operators of any failure in both the air supply and reduction of pressures. So this is more detailed. can we assume that room pressure failure or device pressure failure equals an air supply problem, or do we need to monitor the air supply separately? We need a little bit of thought about that. Clearly, there is a much more definitive requirement. Warning signals should not be overridden without some assessment, and a procedure should be available to outline the steps taken when a warning is given. So this is a pre-considered and evaluated procedure that all relevant parties are aware of. Where delays are set to alarms, because there might be a normal delay while a normal door operation, for example, is being undertaken, opening and closing a door for transit, then it is not uncommon to have a delay, but it needs to be a reasonable delay, and it should be documented and justified within the contamination control strategy. Other air pressure differences should be monitored and recorded at regular intervals, clearly giving you a bit more freedom if they are deemed to be non-critical. Now we move on to some more challenging areas, I think, just to understand the detail and the application. Clean room and clean air equipment qualification. So there's now in the document a very clear differentiation between the business of qualifying a clean room or clean air device and then monitoring it in operation. So the document certainly improved from that perspective. So there's a new section 423 onwards that improves current practice but has some very important changes that I will highlight. It relates Annex 15 qualification tests but lists specific ones relevant to sterile products manufacture. So it lists a whole range of clean room, clean air device related performance attributes, and these would all agree and align pretty well with the similar guidance in EN ISO 14644 parts 1, 2 and 3, particularly part 3 related to testing clean rooms, where you will find some detailed methodologies for most of these attribute tests. Remember that GMP doesn't give you a recipe or a best practice for testing aspects of performance. The GMPs assume there is good engineering practice knowledge and or references to relevant standards, clean room related standards. So we have a new table 1, and this relates to airborne particles for classification. And now we have a big change. So for grade A, at rest and in operation, and grade B, at rest, there is no specification or level or limit of 5 micron and greater particles. And when we look further and drill down to notes A, we find some slightly more difficult language to understand. So first of all, it says classification including 5 micron particles may be considered where indicated by the contamination control strategy or historic trends. So let's assume I'm starting now to classify a new installation where I have no historic trends. Then I've got to decide whether during classification I'm going to consider 5 micron and greater particles or not. It is unclear. Now, in the first draft version of this for public comment, the one that was developed principally under the leadership, rapporteurship of Andrew Hopkins from MHRA, he informed certainly us in the UK that the primary reason for removing 5 micron levels from this table was because in the ISO cleanroom standard it is deemed to be unsafe to measure 5 micron particles and greater at such low concentrations, that the uncertainty of measurement means that it's unlikely to be a robust test. And also he saw that many companies, because they were driven by the 20 and 29 particle limits in the 2008 version, ended up with very large sample sizes and therefore long testing time, and the firms were reluctant to do more rigorous investigation of their grade A zones because it just took too much time and money. And he believed by removing that limit, thereby leading to smaller sample size requirement, firms would easily justify doing much more rigorous classification testing of critical zones. Anyway, so that was the original background that I understood. It's just how would you interpret clause note A? We need some supplementary guidance and thinking related to that. Then it goes on for grade D. In operation limits are not predetermined. This is for airborne particles. The manufacturer should establish in operation limits based on risk assessment and routine data where applicable. Now I would suggest that most grade D spaces, and remember the equivalent microbiological levels in operation are 200 colony forming units per cubic meter, that in operation air cleanliness is probably not a relevant parameter for the type of activities that are going on within the room. But clearly new Annex 1 demands a little bit more conscious thought and some conclusion as to why you are going to set a level or not for grade D in operation at both particle sizes. So a bit more complicated, and I think it's a subject area around 5 microns where you should be aware that there is a working group in ISO TC 209 writing a technical report on airborne particle counting practice, and a lot of that is focused on some level of assessment of 5 micron and greater particles. Both these aspects, I think, are going to be tricky to interpret. So for classification, it goes on to say, it refers specifically to ISO 14644 Part 1, and it says the minimum number of sample locations and their positioning can be found in Part 1, but also Part 1 defines the sample size and the basis of sample size based on the particle concentration at the class limit. So I'm just being a bit picky here that the text is a little incomplete. For aseptic processing, this is an important bit, aseptic processing area and the background environment, the grade A and B areas respectively, additional sample locations should be considered, and all critical processing areas such as point of fill and container closure feeder bowls should be evaluated. So the reason this is here is because ISO 14644 Part 1 assumes a very simple rectilinear clean room or clean air device space. It does not attempt to take into account irregular shapes or process complexity. So what they are getting at in this is for the process of classification to consider in much more detail the configuration of the process and the critical control points within that process. So the language here is new and for some may demand more testing locations. So critical processing locations should be determined by documented risk assessment. Well, that's fine, but the key to it is knowledge of the process and the process operations. That's really the key. Common sense to me. Okay, Clause 5 has another bit, probably the most controversial bit, about particle counter, tube length and configuration. So it's a bit hidden in section 5, and it's all about particle counting. So 5.9 says particle counters including sampling tubing should be qualified. The manufacturer's recommended specification should be considered for tube diameter and bend radii. The tube length should be no longer than 1 meter unless justified, and the number of bends should be minimized. Well, I don't have any problem with minimizing lengths and bends, but we've got to be essentially practical if we want to keep the particle counter away from the critical zone. And the 1 meter dimension has actually been taken slightly, a liberty with it slightly, from a completely different part of the ISO cleanroom standard. So it's the part related to macro particle counting. Portable counters with a short length of sampling tube should be used. Well, I don't have a problem with being as short as possible. Isokinetic, not an argument for unidirectional airflow, and the orientation is not an argument. But it's unclear here whether this applies to both the classification or the real-time particle monitoring, and any sort of qualification of the performance particle recovery efficiency for particle counters is really challenging to do, particularly related to in situ systems. So I think there are some things here that are need very careful consideration. I just reiterate that ISO Technical Committee 209 has a working group that is producing a technical report related to this and other aspects of good particle counting practice. CCN will share as appropriate information related to this as it becomes publicly available. Okay, recovery time, unidirectional airflow velocity. So we still have in the document the 20-minute cleanup time, clause 29, or recovery time. Unidirectional airflow requires airflow visualization, no change to that. And air velocity, we still have this really confusing phrase. Unidirectional airflow systems should provide homogeneous airspeed in the range of 0.36 to 0.54 guidance value at the working position. Now, most of us with clean room testing knowledge and experience realize that we want to choose appropriate average velocity for my clean zone, depending whether it's an isolator, an open traditional clean zone, and so on. So there might be different target velocities chosen, and there has to be a plus minus tolerance on the range of velocities to give us confidence that we're going to have unidirectional airflow. So this still requires major clarification of best practice. Airflow visualization studies should correlate with the airspeed measurement. No problem with that. So it's poorly written and doesn't follow, in my opinion, best practice. So good engineering practice guidance, and guidance that you will find most of in 14644 Part 3, clean room testing, is as follows. Let's assume we have a unidirectional airflow grid over a zone. Could be an open cleanroom application, could be an isolator, could be a RABS. And looking sectionally, this will probably have a plenum of some sort and a number of filters on a grid, a supporting grid structure with gasket or gel type seals. When I'm evaluating the airflow through this, there are a number of things that are important. So first of all, we have the airflow through the filter, and I need to be confident that the air velocity through the filter, reasonably close, maybe 25 to 50 millimeters from the filter, is within the rated range, velocity range of the filter. So when I buy my HEPA filter, it will have been tested by the vendor for a specific grading, let's say H14, at a specific velocity through the core of the media. I need to make sure that my filter in application is not being overrated. If I try and blast too much air through it, the efficiency will decrease and the leakage penetration will increase. I then need to allow the air to straighten up over any obstructions, such as the frame. There may be some light fittings, and indeed some applications have the application of an air distributor screen, sometimes called a CG screen. So I need to determine a test height when the air has straightened out to measure my average velocity, what I call the system velocity. And this might be somewhere between 100 and 300 millimeters from the face of the filter. And I might have a specification ranging typically from 0.45 meters a second plus or minus a range, or 0.35 meters a second would be very common in isolators and restricted access barrier systems, plus or minus a range. I personally set an engineering specification much tighter than the GMP suggested tolerance, but that's a personal preference. Then we come to the working height. Now this is highly problematic. So Annex 1 has this working level phrase, and if you drop down a significant distance from the filter, you probably find you're at the point where the air is flowing over the obstruction and it's impossible to measure a sensible unidirectional velocity, or we actually know that there is a turbulence effect where the velocity measured appears to drop as you move down from the filter. So it's inappropriate to measure the velocity as a primary acceptance criteria a long distance from the filter. There's plenty of good practice such as this, which I believe should be applied sensibly and professionally to address the rather limited and difficult to interpret language in Annex 1. Requalification frequency is much more specific, and it's well aligned with ISO 14644 Part 2, particularly the British Standard version that has guidance in it. The other EN version and ISO versions do not have time guidance in them. So Annex 1 says grades A and B should be requalified 6 monthly and C and D 12 monthly. That would seem to be fairly common practice. Appropriate requalification guidance is given for that, including the things that might trigger additional requalification from change or problematic events in a clean air system, such as interruption of air movement, which affects the operation and so on. Moving to the last few parts, environmental and process monitoring. So this is part of the contamination control strategy, and it's integrated in a whole section on aseptic process simulation in section 9. That is a much more highly developed section. I'm not going to talk about aseptic process simulation today. So there's lots more detail about the parameters that shall be monitored. So environmental monitoring in real time for total particles, environmental and personnel monitoring for microbial contamination, and now there's more specific requirements for temperature and relative humidity, even suggesting that this should be monitored, recorded, alerted and alarmed, even if it is not product specific. And there is lots more about aseptic process simulation, which for many is seen to be part of process validation. So environmental monitoring now has tables 5 and 6. So table 5 for particles and 6 for micro. The micro table is pretty much the same as before, except now we have for grade A an unambiguous no growth. We've got away from that very difficult language about averaging. Grade A and B now have 29 particle limits for 5 micron and greater particles, required to be monitored. So if you remember, the new classification table does not require consideration of 5 micron and greater particles unless you have determined it a requirement in your CCS, whereas monitoring requires them to be considered. So the 5 micron limit is added in table 5, and there are some significant additional notes about the sample size and alert and alarm triggers. So this is quite a complex issue, and CCN will be discussing this and presenting separately on what I call the 5 micron problem. But be aware of this and read the text carefully. You need to have a well-documented, well-defined, with justification monitoring plan. Still missing. I was really surprised at this. Anything meaningful about HEPA filters. So HEPA filters really mission critical in our clean rooms and most critical in unidirectional airflow systems. So in the glossary you will find the term HEPA filter is described. High efficiency particulate air filter specified in accordance with relevant international standards. Okay, doesn't make any specific reference. I'm quite surprised that unlike the clean room standards, it doesn't make reference to the available international standards for HEPA filters. 867 says depyrogenation tunnel, all air supplied to the tunnel should pass through at least a HEPA filter, and periodic tests at least biannually should be performed to demonstrate filter integrity. And then there's a similar thing for dry heat depyrogenation ovens, but nothing about the actual grade and leak test requirements in clean rooms. US FDA's 2004 aseptic processing guidance says a huge amount more. So we are reliant really on current industry practice, and I would say I most commonly see for grades A, B and C the use of an H14 following EN 1822 or ISO 29463. And most firms set a PAO, polyalpha olefin, aerosol leak test acceptance criteria of less than 0.01% when the filter is tested and challenged. Some parties prefer to use a particle counter test method, which would be equally acceptable. So all this is missing from the Annex 1 document, and I'm just surprised about that. I know in various submissions for consideration there were proposals for language similar to that in the US FDA's 2004 aseptic guidance. Finally, to conclude, a suggested approach to dealing with Annex 1. So don't panic. Remember, we've got 12 months to comply for most of the changes and 24 months related to the sterilisation of lyophilizers and other specific freeze-dryer practice. I would recommend that you do a gap analysis pretty quickly with existing practices and identify changes and improvements that you might need to make, including your contamination control strategy, and then implement. And just remember that guidance, GMP guidance like Annex 1, relies on supporting standards and good engineering practice. It's never going to be a specification of detailed technical solutions or detailed testing and commissioning practice. We have to invoke relevant expertise and other reference standards and practices. So I hope that has been useful as an introduction to the clean room related aspects. I've been very open about areas of concern. They are somewhat a personal perspective, but also reflect commentary and discussion that's already been fed back into organisations like PHSS here in the UK, ISPE and PDA in a more international sense. Okay, well with that I'm going to close the meeting, and I'd like to thank you all mostly for your attendance. Really appreciated. We had virtually 100 people, just short of that, which was really good. So thank you very much. I hope you found it useful, and I look forward to seeing you again in the future.